The adapter hypothesis was proposed by Francis Crick to explain how DNA is used to create proteins with a chain of specific amino acid sequences. He proposed that an adapter that binds to a template strand is able to translate it from base pairing. From this, the adapter was able to recognize specific nucleotide sequences that encoded an amino acid. This allows amino acids to bind together into a specific sequence to form a protein based on the order of nucleotides in the strand. The molecule he called an adapter was later recognized as transfer RNA, or tRNA, which transfers the mRNA strand based on 3 nucleotide sequences called codons into a specific order of amino acids. This process is known as translation. At the beginning of translation, we have a ribosome that binds to the mRNA and moves into the area of the mRNA strand where translation will occur. A tRNA molecule will come in and bind to the part of the mRNA codon that is complementary to its sequence. Another tRNA molecule will come in and bind next to the mRNA codon. Both tRNA have amino acids attached that are coded from each respective codon. The ribosome then acts as a ribozyme that transfers the first amino acid into the second one and forms a peptide bond that can connect these amino acids together. Then another tRNA binds next to the codon, and this continues until a long chain of amino acids forms. The order of amino acids is determined by the order of mRNA codons in the template strand. By definition, an adapter is something that can be used to connect things that normally cannot be connected directly. So since amino acids cannot bind to an mRNA strand directly to know the order for them to form proteins, tRNA has to be used to recognize these mRNA codons and convert them into proteins. From this, the tRNA acts in the same way as Francis Crick's adapter. However, if tRNA poses some alterations, such as an RNA mutation, this could affect the protein that is formed by the amino acids. For example, in a nonsense mutation, this changes a codon that was an amino acid into a stop codon, which could result in a protein that has less amino acids than needed, forming a non-functioning protein. Another alteration could be a read-through mutation, which changes a stop codon into an amino acid codon. This leads to the protein having more amino acids needed, which also forms a protein that can no longer function. These are my sources, and thank you for watching.